semester. So short that I don't have separate slides and study guide. It's all in one. Study guide is the slides. So this chapter covers all 5 special senses. I know that you will read this chapter very carefully over winter break as pleasure reading. But for now, for us, we are just going to talk about taste, smell, vision. Not going to touch the ear with equilibrium and hearing. And for taste and smell, it's going to be very limited. A couple slides. And then the rest of it's vision. And we are doing an eye dissection, cow eye dissection, at the end of class. So the objectives differentiate between general and special senses. And we've talked a lot about this already. Identify the special senses organs of olfaction, gustation, and vision. So there's a little bit of anatomy. This will be on the clickers portion of the final. Describe the internal structures of the eye, more anatomy. Explain their functions. Differentiate between rods and cones. What type of receptor is the rod and cone? Photoreceptor. Last, describe losses in the visual field due to lesions, which is damage, to different parts of the visual pathway. And that last objective pertains to the last slide. So in total, 5 special senses. Touch would be an example of a general sense, right? When we're comparing special senses to general senses. One big difference, general senses receptors are widely distributed. Where do you find Pacinian corpuscles, for example? Everywhere. In the skin, in joints, in the ventral body cavity, everywhere. Widespread. Special senses receptors, also called special senses organs, because it's typically multiple cells working together as the receptor. These are not widely distributed. They're very localized. Where do you find all photoreceptors? In the retina, the back of the eye. It turns out 70% of all receptors in the body are photoreceptors. But they're found in 2 very small places, right? The back of each eye, very localized. How about olfactory receptors? Roof of the nasal cavity as the olfactory epithelium. Vestibular apparatus and cochlea. Ear. Mm-hmm. Taste receptors. They're in the taste buds of the tongue and the pharynx. And in the pharynx or throat on the epiglottis as well. And that's what vagus is involved with. Providing taste inputs from the taste buds on the epiglottis. It has nothing to do with taste from the tongue. That's just facial and glossopharyngeal. So we'll look at gustation and then olfaction and then vision. As far as receptor type, we're dealing with chemoreceptors, and that's true for taste and smell. So transduction isn't that difficult to understand. It's very similar to mechanoreceptors. Instead of distorting the membrane to open a mechanically gated ion channel, now we just use a chemical that binds to essentially a ligand-gated or chemically gated channel. The channel opens, sodium influx, depolarize the receptor membrane. If the graded potential is large enough, reaches the initial segment of the axon, and you've got action potentials. Pretty straightforward. So taste and smell, not so bad in terms of transduction. But the other 3 special senses, much more complicated. All covered in the chapter reading, if you're interested. The taste receptors, which are chemoreceptors, are housed in taste buds. which are primarily on the tongue. Turns out there's 5 basic tastes, different categories of chemicals that provide us with different sensations of taste. There's the sweet, sour, salty, bitter, and those are the 4 classic tastes. And more recently, in the last 10, 15 years, a 5th one has been added, umami. And umami was discovered by a Japanese scientist, hence the name. Sweet tastants include sugars, alcohols, some amino acids, like aspartame, and artificial sweeteners. And that's what artificial sweeteners are, non-sugar chemicals that stimulate the sweet receptors. So it's as if we are tasting sugar. We can't tell the difference. Anything sour really means anything acidic. So these are almost like pH receptors. So citric acid, which is found in fruit, provides that sour taste, that tart taste, like a raspberry. Salty, inorganic salts stimulate salty taste receptors. Sodium chloride is the most common and the strongest of the different salt stimuli. Bitter tastants include alkaloids and some non-alkaloids. For the most part, medications in the form of pills are largely alkaloids or non-alkaloids that stimulate bitter taste receptors. So if you put a Tylenol or an ibuprofen or an naproxen or pretty much anything on your tongue, you let it dissolve a little bit, doesn't taste so good, it's usually kind of bitter. That's why. Umami can best be described as the beef taste in a steak. It's a beef flavor. The artificial version of this is monosodium glutamate. And so this has long been added to our foods to enhance the flavor, the taste. MSG. Gustatory pathway. 3-neuron chain. This one's probably the most similar to somatic sensory pathways, which also use a 3-neuron chain. Neuron 1 takes us from the receptor to brainstem nuclei, 2 from brainstem nuclei to thalamus, 3 from thalamus to cerebral cortex. So the taste receptor cells, or taste cells, have really long axons. They extend from the taste bud all the way to the solitary nucleus, or NTS. When we looked at NTS in chapter 12, what function did we associate it with? What type of sensory input did it receive? Because at that point, we didn't talk about taste. Was it somatic sensory input or visceral sensory input that went to the NTS? Somatic went to nucleus cuneatus and gracilis. It was visceral. But interesting that not only does it receive visceral information we don't become aware of, it's also receiving taste. Certainly different parts of the NTS are receiving these different types of inputs. And then from NTS, projection fibers take this information to the thalamus, the VPN, and then from the thalamus to the gustatory cortex, which is located in which lobe? Insula. Where does crossing over occur? Brainstem. You can see when the second order neurons leave NTS, they cross over to the opposite side. So remember, everything crosses over. I was going to say except, but I can't do it that way. Everything that we're aware of crosses over. Information going to the cerebral cortex, where we perceive the sensory input. That information crosses over. What I was meaning to say, everything goes through the thalamus except what? olfaction. So even taste goes through the thalamus. Olfaction was the one pathway that doesn't. And because of that, we don't need a third order neuron, just two. So we're bypassing the thalamus. So next is olfaction. Like gustation uses chemoreceptors. Also like gustation, like tastings. Odorants have to dissolve in water before they can stimulate their receptors. Right? So chemicals that stimulate taste receptors can be called tastings. They have to dissolve in the saliva in your mouth in order to diffuse into and stimulate their receptors, which are buried inside of the taste buds. Same is true for olfactants. Olfactants are chemicals that are airborne. Right? So they come into your nasal cavity, they reach the olfactory epithelium, they make contact with the mucus lining the olfactory epithelium and dissolve in the water. If your nose is dried out, right, if there's really no moisture in that olfactory epithelium, you don't smell so good. You have a real tough time smelling. A simple test trick, really, you could do, you can stick out your tongue, take a paper towel, blot it dry, completely dry, no moisture on your tongue, and then put something flavorful like, what is that stuff, the candy Fun Dip, where you have different flavored powders. It's sugar, essentially. You could do just sugar. Put sugar on your tongue. You can't taste sweet. You can't taste anything. If your tongue is completely dry and that sugar doesn't dissolve in water, you won't taste it. And we also know that taste and olfaction are connected. It changes our perception of taste when our olfactory pathway isn't working very well, like when we're sick and congested, or anything that affects our ability to smell, it in turn affects our ability to taste. We need both. So the olfactory pathway doesn't go through thalamus, doesn't need that third neuron, so it's a 2-neuron pathway. Neuron 1 is the olfactory receptor cell. It's a bipolar cell. So at the end of one process, which would be a big dendrite, you have dendritic endings that are called olfactory cilia, and they're in the mucus. And then the olfactant comes in, gets in that mucus, dissolves in the water, and diffuses and reaches the cilia and stimulates those chemoreceptors. And then the information travels through the dendrite, at this point as a graded potential, hits the initial segment of the axon, becomes an action potential. And then the axon goes through the olfactory foramina, through the cribriform plate, into the olfactory bulb. These axons are actually what we refer to as cranial nerve 1. That is the olfactory nerve. But they're not all bundled together like a typical nerve. Instead, we just have nerve filaments, olfactory nerve filaments that are going through the olfactory foramina. In the olfactory bulb, which is this big structure here, we have mitral cells. That's neuron 2. That's your second order neuron. So the synapse between first and second is in the bulb. The mitral cells then use their axons to transmit these action potentials all the way to the olfactory cortex. So that's it for taste and smell. Last is vision. 70% of all sensory receptors in the body are in the eye. That's a lot of information to process. Just your visual input alone, coming from 70% of all your sensory receptors. So not surprisingly, about half of your brain is involved in processing visual information. And when I say half of your brain, I'm referring specifically to the cerebral cortex, but that doesn't count for a huge amount of your brain surface area. Now, that being the case, if you are born without the ability to see, maybe something is wrong with the eyes, optic nerve, whatever the case, if there's no visual information coming into the cerebral cortex, that's a lot of area that could be used for something else. And luckily, the brain is plastic. Neurons can take on new functions, if need be. So then it does kind of make sense how people that are blind have the ability to use their other special senses in a way, or general senses as well, in a way that we cannot, that we can't even really imagine. I mean, I just can't imagine reading Braille, for one thing. Right? The tactile information that they're gathering from these little bumps and crevices, that's incredible. They can actually read based on touch. Well, their visual cortex is processing a lot of this information. It's not just the somatosensory cortex, the visual cortex is getting involved. We're going to use that surface area. We know that blind individuals can learn to use echolocation to navigate, making little noises and listening to those noises bounce off of objects. So just like many animals use echolocation, humans have the ability to do this as well. Question?
Oh, no, I was just going to say that, like, the people that click, they basically say that it's almost the same as seeing, it's just
Yeah.
color range. They
Right.
get the same thing.
And the information is auditory in nature, but the visual cortex is involved in processing it. So they see, in their mind, they see, not like we see, but they have a map. That's how they're able to navigate, by listening to these sounds. It's pretty crazy. Eye structure. So you have a wall, and then you have a hollow interior. Right? So the wall of the eye has 3 layers, and then the inside, which is where you have this huge open space, filled with fluid. So we'll look at the wall first, after break.